Good morning. I have a double duty. The first one is to talk about clinical trials in general, to give you a sort of background, how these particular drug trials, maybe in the future will be other type of intervention, perhaps in combination with drugs, are done. And I'm going to mention briefly where we are with the IGF-1 trial, which is, as you heard, coming to an end in terms of recruitment. So why do we need to do this? I mean, this is the big question. Why do these trials have to be done? Because we need to make sure that the drugs are effective. We collect a lot of information, these anecdotes and observations. Some of us have a better gestalt to recognize patterns when we see patients in clinic. But there's no other way that you cannot ensure that there is a story there. There is a really advantage of a drug until you do it. And they can be done with new drugs. I mean, we have put a lot of emphasis on new drugs, but it can be done with drugs that have been around for a while for other indications. It could be a different disorder. We need to make sure that the drugs are safe. And assuming that the drug is safe in adults who develop what we call neurotypical is going to be safe in Rett syndrome, it's not adequate. We need to make sure that the drugs, particularly the novel ones, are safe. We need also to make sure that the drugs are easy to administer. If the drug had to be, the schedule of administration is so complicated that it really is incompatible with domestic life, obviously that's not good. I am a clinician, so I'm interested in doing research, understanding the mechanism, but ultimately, if I am embarked in a clinical trial, I want to make sure that the outcome of that clinical trial, if everything is successful, is a drug that the FDA is going to approve. Why is it so important the FDA and in Europe the EMA approve a drug? Because drugs are made by companies, and the companies are not going to be invested in producing the drug, and some of them are very complicated to be made. It's a very expensive undertaking without a guarantee that the drug has a sort of a seal of approval. So we need to make sure of that, and also we need to make sure that the drug is affordable. We don't want to prescribe a drug that is really you had to mortgage your house. Really, the drug had to be affordable. In the United States, this is through prescription plans. Even in Europe, which is mainly governmental, the governments really want to make sure that the drug is worth supporting. There are more reasons. So why are they needed? Because even though you can go to sessions, and I don't know how many of you went to the sessions earlier, and there's a lot of excitement, an animal model. This morning, I got an email from the administration of our hospital. They always, when there's something happening in autism, they send me an email, do you want to say something that is on our website? Because there was another press release from an institution showing that the drug, an old drug, now is effective in autism because they tested a particular animal model. We are bombarded with the information. To be honest, we are in academia. Academic institutions like to do that because they look good and they keep the momentum, a positive momentum. But from the animal model to the patient, there's a big distance. The reality is that even though in an animal model, a drug could be good at rescuing all the phenotype, what we call phenotype, all the manifestation, it may not be in humans. And that's something that we have learned already in Fragile X syndrome. So we need to make sure that we know what the drug is being effective for. In theory, it's going to correct all neurologic problems. In practice, it doesn't. And the next is that we don't know if the patients are going to tolerate the drug well. So going back to the issue of safety. So clinical trials are developed in stages, and you may have seen this actually now. Schemes like these are very common. They present in multiple lectures. It's a long process. It's an expensive one, and it's one that goes from the preclinical side, and you have the time frame, all the way to the phase 3. The phase 3 trials are the trials that, if they are successful, the data is presented to the FDA. We're very concerned about how to accelerate this process, but the reality is that the process in some way had to follow all these steps. It's very difficult to have shortcuts. Phase 1 trials are to look at safety. It's the first look at the drug. Is the drug safe? Can we administer the drug? Can it reach the right levels in blood and in the brain, in our case? The phase 2 trials, which you're going to hear about, are the trials that are called response or efficacy trial. It's the first time that we are really monitoring that the drug is doing something in terms of correcting symptoms. Now, in order to accelerate the process of drug discovery, we are thinking about different ways in which phase 1 and 2 trials are combined to take advantage of what is the early stages of the trial, and we can accelerate, we can go to the phase 3 as quickly as possible. And this is a very fluid field. There are many new ideas. Some of that, the lead has been taken by oncology. I place there also the number of patients that typically are done in a trial, like let's say for diabetes, for hypertension. We're dealing with a rare disease, so we're trying to deal with fewer numbers, not 20 to 80 for a phase 1. Our phase 1 was 12 subjects. For phase 2, we don't have the capacity to do hundreds, so we're trying to do a number that has enough statistical power to answer the question. And ultimately, the drug is submitted, the data on the drug is submitted to the FDA. The FDA is saying everything okay, and then there is still a continuous process of checking whether the drug is safe. We have had bad experiences. Vioxx is probably the most famous one. It was approved by the FDA, and data on cardiac side effects was discovered later and is now off the market. There are many designs to do this, and each one has drawbacks. Crossover designs are very nice because everyone gets a drug, randomized, whether in the first half of the trial or in the second half. So that's a good thing, but those are very long trials, and many things can happen. I had to tell you, because we're doing a crossover design trial, many of our girls have had seizures getting worse or new onset seizures, and we are not going to stop, administering the current treatment for seizures if they cannot be controlled by the drug. We don't know if they're getting the drug or the placebo. So a lot of things can happen, and the data at the end can be difficult to interpret because there are so many other factors that can affect the outcome. The parallel design, which is the most common of all designs, is a problematic one for many developmental disorders because there's no guarantee that the patient is going to get the drug. So some fields like Fragile X have decided that we're going to do all trials with an open label extension. We are going to demand from the drug companies that they have to give us the drug at the end, so those patients who respond, they have drug after the trial, and the drug company can keep collecting data. But that means that the company had to agree on doing that. How do we measure that the drug really makes a difference? We can do very fancy things. We can do fancy electroencephalographic recordings. We can do complicated measures of all type. But at the end, we need to make sure that these are functional and improve the quality of life. To be honest, the FDA is a little bit behind. They are still very much in the era of pencil and paper, and they are moving slowly, and I think that we are hopeful they're moving slowly to more technologically derived ways to demonstrate that the drug is effective. But the bottom line that the drug has to affect and improve the quality of life is a requirement. There have been a number of experiences in which there are some incremental changes, very nice, statistically significant, but they are really not, they don't make a dent in the problem. The quality of life is not improved. We need to try to look at as much objective outcome measures as possible. And we hopefully will have to support the industry. I had to say in our IGF-1 trial, we had to make placebo. It's something that I don't want to go through again. We became manufacturers of placebo. I am a laboratory person. I know a lot about how to work with cultures. I never have had standards of purity, industry standards. We are not in that business. All that is avoided if you work with industry. So, closing this first part, I want to make emphasis on some issues. And I know that this is a very emotional one. A clinical trial is not a guaranteed treatment. It's a research project with a treatment being tested. So I know how many families really want to be enrolled in trials. And that's the reason why we have come up with this idea of designs in which everyone gets the drug. But they are not easy, and they come with a cost. It may not work. maybe the drug may not be safe. The problem is that if the trials last very long, you may not know that it's a failure for a period of time, and failures have very negative consequences. The mood from being very, very bright goes to be very depressed. And I think it's difficult. It's easy for me to tell you, try to stay cool, but the reality is that emotions play a big role. So we need to accelerate trials not only because you want the drug soon, because you want to know the failure soon. So I think the very emphasis on, for those of us who were present at previous sessions at the meeting, a big emphasis from the FDA and NIH and from the clinical trial communities to do many of the early phase trials very fast and to know whether there's a failure or success. And failures coming soon is what we want. We don't want failure after years. They are complex and expensive, and I know that this is what makes the foundation suffer. It's very important the trial cannot poorly design, and I think that this is, some tension among ourselves. The design had to provide an answer. If at the end of the day you cannot tell whether the drug worked or didn't work, it's terrible. You spend all that time and all that money without an answer. It's better to know that it doesn't work conclusively rather than a maybe. And the real clinical trial required a team and infrastructure. The natural history study was the first topic of this session. Natural history studies are very important because even though we have a placebo, in our trial there is, and we really need to build a control in trials, but they may not be sufficient to understand whether a drug is effective. We need to see what happened. What will happen if this girl didn't get anything? And that is come from looking at natural history studies. There's a big emphasis from the NIH and FDA on doing that. And we need to work a lot on outcome measures. So if one day you hear that the foundation, RettSyndrome.org is funding an outcome measure, say, well, they are not funding the development of a drug. Please understand, we need to get good outcome measures. Otherwise, we cannot measure whether a drug is effective. They require a lot of planning, and people are very impatient, including ourselves, because we want to see things happening. Now, I wanted to touch on an issue that I didn't touch. I gave this talk last year in Utah because it's an issue that we have experienced, and I have experienced also as a Fragile X researcher, social media. You can do a lot of good things with social media, and you can do a lot of bad things. That's the reason why I'm not on Facebook. I am not on Twitter, because it happened in the Fragile X community. Parents talk to each other about how come up with a score on measures to be eligible. And so they could get on the trial. The trials had a lot of problems because of that. If someone experiences a side effect and the parent is angry and broadcasts to the rest of the world that there are side effects, it may have been in one situation, creates bad momentum. So please, be very cautious about what you say. These are days in which we all have a tremendous amount of power because we can broadcast to the entire world. And ultimately, I wanted to say this because if social media is something that I brought to your attention at risk of you getting angry at me, is because you have to understand this is a true partnership. And this sounds like sometimes like you are saying the partnership. This is true partnership. All of us play a role in doing this, and you play a very important role. The foundation plays a role. Our team, I don't know if the members of my team really, I think they left, but they are critical. I couldn't do this without them, and those of you who have been in Boston, you know how valuable and they are. It was mentioned in the opening talk how good was the experience on going to UAB. All of that is because there is an entire team. We are all committed to this. We all have the same purpose, to get an answer, and to get an answer soon and the right way. Shortcuts are happening in the short term, but are happening in the long one. So I am going to skip this, and I'm going to give you an update. Sorry for going very fast, because we have a lot to cover, but I wanted to give you an update. So we finished a phase 1 trial. Actually, I'm sorry, I didn't update. The publication is out. It was published at the end of March. So if someone is interested, I can provide the publication. I'm not allowed to give it to everyone for copyright issues. But the bottom line is that it finished in 2012. The paper came out after a very long review in March. The drug is safe. It was well tolerated, even though there are 2 daily subcutaneous injections. The parents did a terrific job, no complications, and it was effective to treat anxiety and apnea, breath holding. We wish it would have been effective in treating everything, but again, this was an open label trial, so in that regard, so we may have discovered something that is not going to be proved in the phase 2, but we may have missed something because it was a small number. There were only, at the end in an open label extension, were only 10 girls. Now we're doing it, and we are about to complete enrollment on a phase 2 trial. This is the trial in which, despite still monitoring safety and tolerability, is about efficacy. This is where we're testing whether the drug does something good. And it's a crossover design, so everyone gets the drug, but randomized. I have no idea who is on what because it's done by other people. All the safeguards have been put there to be a really blind study. 30 girls will start in March 2013. Because we have a long problem with production of placebo, we're not going to end until about late fall 2015, because we need to keep the same so-called washout period, the period between the 2 phases, constant. And we are targeting the symptom that was shown to be effective in the phase 1, but we're collecting data in case that there are other positive effects. So we're not going to miss motor improvements, cognitive improvements, and all of that is really being collected, as well as quality of life, which is very, very important. And this is to show you, the top one is the diagram of the phase 1, which ended and was published, and the bottom one is the phase 2. You see half of the girls are randomized to the drug, and half to a placebo, and then they cross. That's the reason called crossover, and this is a period in which they are not on the drug to make sure that they come to the second segment, to the second stage, they come without too much effect of the drug. One thing that we're able to do on the basis of the phase 1 is that we got authorization from our IRB to prescribe the drug to every girl who show a positive response during the phase 2. I cannot guarantee that your prescription plan is going to pay for that. We will have to make arrangements for someone who knows how to manage the drug, because most of the girls are not from Boston, so make sure that someone somewhere else can do this. I am going to skip all this to give you a brief review of where we are. This is the inclusion criteria. We changed to 2 to 10. Everything else has remained the same. Obviously, we are treating anxiety and avoidance. It has to be, the girl had to have some level of problem in that area. I mean, if you recruit someone who doesn't have the problem and you're treating a problem, you are going to fail simply because you selected the wrong population. So there are some inclusion criteria that are very logical based on what we're trying to target. And there are exclusion criteria also because we're given a hormone that can affect growth, can affect puberty, so we need to be very mindful that we don't want to accelerate that. I know there's a lot of information. If you are interested, you can approach me later, and I can provide more details. And this is the final where we are. We have pre-screened 53 girls, and we have essentially most of the cohort selected. So we have 23 are already in the process. We need 7 more to start or to complete the first stages. So we are almost there, and we screen a large number because the way that we do it is to pre-screen without the family having to come to Boston to ensure, to optimize having a positive screening at the moment when they are on site. So we're doing well, and all this has been thanks to everyone's collaboration. And there are some openings, but not many. There is always a possibility that some of the girls that still have not begun, not because they are not in the process, because we have a limited capacity to enroll, to get girls through the system. Some of them may fail, so that opens the possibility to a few more recruitments. And That's our email. So if you have any question, Rett Research at Children's Harvard EDU, and those are the numbers. Thank you.
So while my slide is coming up, I just want to say I took Mickey's advice and I wore platform shoes also, so I could see over the podium. There's one other thing I want to say is that I have to get on a cab at 12:45, and so I'm going to go through my slides actually pretty fast. And I wanted to apologize to Dr. Djukic and Dr. Naidu if I have to run out because when you guys are still talking beforehand. But I'm going to go through it fast, and maybe if there's some real burning questions that people have, well, I can try to answer those a little preemptively before the end, just so I can get through that. Okay. So these are just my disclosures. I think it's important just to give disclosures. You know, I think we all know the basic work that was done in Mrganka Sir's lab to establish that this IGF-1 tripeptide, the first bit of this protein, seems to help the mice. I'm not going to go through this data. I think we're all familiar with this, that it helped the mice live longer, breathe better, move better. Neuron Pharmaceuticals, which we've heard about today about Larry Glass 2 years ago announcing this, they had a compound that's a methylated version of this tripeptide of IGF-1, so it has just a little extra chemical bit, and that makes it stable. It has better pharmacological properties, and importantly for us, it's able to be taken by mouth. The other thing is that this is something that has neuroprotective properties and anti-seizure properties, and for that reason, it's been in clinical trials funded through the Department of Defense for traumatic brain injury. Sorry, TBI is traumatic brain injury. And because of that, they have a lot of safety data on adults. So that is part of the reason why when we decided to do a trial, it was started in adults, because the required information needed for the FDA to bring this into people, they have all the data for adults, not for children yet. This drug now is also in clinical trials for Fragile X because they did a variety of preclinical work showing that it improved the animals, and that's ongoing. So I think I'm just going to quickly mention the design of this trial. It's called a 2A, phase 2A, as Walter kind of alluded to. That's where we're looking. In this case, we're looking mostly at safety, but we are looking for signals of efficacy. And this is a double-blind, placebo-controlled 2-dose level trials. We had a first cohort at a low dose and a second cohort at a high dose. We ultimately wanted to have at least 48 people. We're actually going to be exceeding that. The ultimate end will be 54 people, with 27 in each cohort, but the goal originally was 48. I listed here how the randomization processes and the age range, and part of you have to have typical or classic Rett syndrome, and you have to have a mutation in MECP2. And this has been a source of consternation as we've been going through the trial for some people, and you know, I can explain that to people if they want. And as I mentioned, we're looking at safety and then a lot of signals of efficacy, really with the goal of this being the first shot to cast a wide net, so that if we find evidence of efficacy in the second stage, we can target that. Very similar to what Walter described. In their first phase, they found it seemed to help breathing and anxiety, and so in the second phase, that's really where the targeting is. And that's how drug development really goes. Just mentioned these important exclusion criteria, which I think we've covered, but I think it again comes to a certain point of consternation with having been basically on any other experimental drugs and having a long QT. And that's actually with the heart rhythm abnormality. That's a fairly common exclusion for pretty much all drug studies, because a lot of drugs can induce this. So we started at Texas Children's Hospital in April, over the summer of April of last year, April 2013, over the summer of last summer. University of Alabama, Birmingham, with Dr. Percy and Jane Lane came on. And then in the beginning of this year, Gillette Children's Hospital in St. Paul, Minnesota joined us. This is just a really rough, I'm just going to go through this pretty fast, which is the series of events. Essentially, people come for a screening visit about a month beforehand, and we establish that they're eligible. During this month, then at home, they record their seizures and their medications. Then they're admitted to the hospital for basically a 5-day admission. And during that time, we have a baseline, what we call the baseline visit, where we perform extensive characterization of your daughter. And then for the next 5 days, they're escalated up into the, the drug or the placebo. It's randomized, and we don't know what it is, and receive, again, continual monitoring for these 5 days in the hospital. Then going home, again, doing the seizure diary, coming back on day 14 for an evaluation, and day 26 is the final day of full dose of drug. Then we down titrate for 2 days, and day 28 is the last day on drug. Now, some people have asked, why 28 days? Why not a month? Why not 30 days? Why not 10 weeks? Why not much longer? And again, this is dictated by what the FDA will let us do. They strictly will only let us do currently 28 days of exposure. And finally, then we have basically a little more in the 2-week washout period when we see the subject again at the last day, we call day 40, basically to look to see if there's a change back to baseline after having been on drug or placebo, or if there's what we call a carryover effect. So we completed that first cohort in January of 2014. That was the low dose or the placebo. We had no serious drug-related adverse events, and that's important. So what's called the Data Safety Monitoring Board then allowed us to go to the next. We got the green light to move to the next stage, which started in January, and the goal is to complete that in the fall of 2014. So this is the announcement that was put up yesterday from NERN. And what this is besides having a typo I see in the title, enrollment usually has 2 L's, but this is an announcement saying that we have completed enrollment. So yesterday, or I think it was Wednesday, was the last person started the trial. And that means they have what I outlined to you, which is essentially a 2-month arc. So the goal is to then them be done about mid-September. So really, when I go back here, let's see if I have another slide. No, I stopped there. So I'll just let you say. So the goal is we're going to complete these last set of people by the fall. And then during the fall, well, it has what's called a database lock. That means all the information gets entered and it gets closed down. And then the code is broken and the data is analyzed. And so the plan is to be analyzing that data this fall and have what's called top-level results in end of, by December, to be able to present to the FDA in the beginning of 2015. So, there's 2, I'll make my acknowledgements and then I'm just gonna make a few comments. This is, really a hard set of work by a large number of people at Texas Children's Hospital with myself and Dr. Glaze, and we have a great physician assistant, Tui Din, and a great research coordinator and project manager, Aaron Knight, and University of Alabama, Birmingham. Of course, Alan and Jane, and they've worked very hard. And then we had the pleasure of adding on Gillette Children's Hospital with Arthur Basang and Timothy Fenya. And I think people know some of the people at NERN. Now, a couple comments I wanted to make, which is, one is people always want to know about a pediatric trial. I think that's maybe where Mickey was getting to in terms of age and what is going to need to be done before a pediatric trial occurs is that the drug company is going to have to do safety in juvenile animals. The FDA requires, before you bring it to children, that additional sets of safety data and what's called preclinical toxicology occurs. And so that's still something that has to occur before that happens. You just have to hold on, and we all are very interested and eager to move, to try to bring this into a younger generation. The other thing, and something just to elaborate on something Walter was getting at about why you do clinical trials, and I think an important thing, and people here have heard me say this before, and Mickey kind of alluded to it, is that you have to remember that a clinical trial is an experiment, and we're experimenting on your daughter. And so for that reason, you shouldn't be upset if we say you don't quite meet our inclusion criteria or exclusion, because they're there to be safe, right? They're there to keep your children safe, and that's why it's set up like that. The other thing is, trials are not there to provide drug for free or provide drug that isn't really available yet. It's an experiment to find out if these drugs work. And we have to find out, we have to do the formalism to find out if they work, because otherwise we're just wasting our time. So that's all I really wanted to comment on those things. If I could just take, if there's one or two questions in case I have to run out, I'll take them.
If the child has the exclusion criteria but doesn't qualify for a trial, what happens if the drug is approved? Would that child be disqualified from taking that drug at that point?
That's a very good question. So the question was, if someone is excluded from the clinical trial, but then the drug goes through the FDA and gets approved, does that mean they would not be able to take the drug? So that's probably not the case. This is the exclusion criteria deal with the trial. It doesn't deal with what an approval, the actual indication and what the FDA will regulate. Now, because also after a drug is approved, it's the physician that decides to write the prescription and give the drug. So in that situation, let's say that we were concerned about long QT, and let's say you're doing a clinical trial and it does lengthen QT, the drug, because lots of drugs do that, then it might come out with it might have an indication for treatment of Rett syndrome, but have a warning that this drug increases long QT. Now, that doesn't mean that a physician cannot prescribe that drug. It just means that it's up to the physician that they're responsible now for screening to see if you have a long QT and making a clinical decision if the risks outweigh the benefits to give this drug that might lengthen the QT in your child, right? So that's sort of the thing. Now, the one thing I might say is that if we say the inclusion criteria is that you have Rett syndrome, and then we get an indication, if the indication is for treatment of Rett syndrome, if you then try to give the drug, if a physician gives the drug for some other disorder, not Rett syndrome, then physicians can prescribe that, but third-party payers oftentimes won't pay for it. So, and that actually kind of goes to what Walter was saying about getting the opportunity to prescribe IGF-1. Well, IGF-1's expensive, and so a lot of insurance companies and stuff might balk at doing it. Now, if you get an indication, it's very difficult for the third parties to try to balk at that. Okay, one more question, and I don't want to take up people's time, but yes. Well, okay, yeah, that's good. So, right now, this is a phase 2A trial, which is safety and signals of efficacy. Typically with a phase 2A trial, that's the first pass, right? And this is showing that it's safe. And in a typical fashion, then the FDA gives an approval to go on to a next level trial, which may be a phase 2B, which may be showing responsiveness in a larger group of people. Instead of being primarily focused on safety, it will focus on efficacy. Or even what we call a phase 3 trial. Now, it's not that there's a pathway of development that is more clearly worked out for common diseases. And the FDA does recognize for rare diseases, or orphan diseases as they call them, that you don't have phase 3 trial. You're not going to be able to have a phase 3 trial that'll have 20,000 participants, which is what might happen in a depression trial or something. It's a different bar. They recognize you can't have 20,000 subjects. So, what I'm trying to say is that this is the beginning, and so I would not be saying, if we go to the FDA next spring, by next summer we're going to have an approved drug. What might happen is next year we have something gets designed more as a pivotal trial, what would be called a pivotal efficacy trial, where you focused into maybe a few things, like Walter mentioned, how they've done it, where these are where we think things are, the signals really were, and so you do another trial to look at efficacy, and maybe longer, maybe hopefully we'd get more kids, maybe we can go into kids, all of those things. But you do a longer trial. But this is, don't hold me to this. This is speculation at this point. We have to get to the end of the trial, analyze the data, go to the FDA and get advice from the FDA what they would like to see, what they want to see as the next step. Because that's, at this phase, it's sort of an iterative process with the FDA. You have these conversations, okay, and they'll say, well, this is how we would like to see what we would feel good about for our next step, so we would think this would be ready for indication. Okay? So I'll stop to let the next people speak. Thanks.